所以这是我想抛砖引玉的，就是引起大家注意这个现象，就是就是有客观原因，有这个国际，就像邓小平那个时候评论六四，说有国际的大趋势的原因，就是中国和这个西方的交恶，这是一个原因。还有更大的原因是我们内在的原因，就是我们这几十年培养的人的价值导向和我们先辈们的价值导向是有很大的区别，所以外在的和内在的加到一起，可能是促成了，就是现在我们这个很尴尬的境地，就是我们做，呃，我记得这个谁呀、呃，James Watson 这个曾经这个在一次三面党里面就讽刺中国学生，他说冷泉港的这个 PI 不要过多关注实验室的事情，要关注大方向。You keep a bunch of Chinese cooking，就是这，就是他的这个名言。我当时听了就非常的反感，就是中国人只会做 cooking 的事情，不能去做大师。所以我抛砖引玉，把这个引这个话题引出来。当然我个人这个故事呢，其实一会儿这个做完三面呢，有空呢我再给分享。这里面就是三个因素促成了，就是一个本来原创的东西，到最后连影子都没有了第一是就是说这个东西这就是被别人这个很快把名字改掉了，而且这个人是大牛，这个一改掉以后。像我就没有生硬了，这是第一。第二就是说，在公司做科研是做童养媳，就是公司的新来的领导人说你不可以做基础科研了，你就必须停止，所以就没有再做 ER stress 这个领域的后续。
嗯
第三就是说，这个等我这个就是从公司再跳回大学的时候，这个已经热的太热了。也不符合我个人的科研理念。就这三个因素哦，还有一个第四个，就是说我是第一作者，又是通讯作者，以至于这个 Linda Hanshoff，他是一个 ER stress 大牛，在 St. Jude 的这个 Children Hospital，他在第二年的时候写了一篇 Cell，他说 Ron Weck 和 David Ron 他们共同发现了 p e r kinase。那 Ron Weck 只是一个参与做的，他是 last name 是因为他做了一点点贡献，所以欢迎大家去挖坟。就是说，
嗯
这个中国人经常说的一句就是对，欢迎大家挖坟，就是把这过去这个来龙去脉挖一下。其实这里面就是我是发现一个很奇怪，就是也很狼狈的一个现象，就是14年的纳斯达，它是给了 ER stress 的，但 ER stress 两个最重要的领域，一个 RE1，一个是 PERK，它只给了一半，这一半就是给了 Mori 和 Peter Water，原因就是不知道谁 clone 了 PERK。这个19年，这个 Peter Water 来我做 seminar，我跟他聊起来了我的故事。他说：“哦，I know somebody clone Perk, is that you？” 我说：“Of course。”我说这个，I hope someday when you go to Stockholm, you would acknowledge our comfort, contribution. 他说：“Oh, I hope we go together.” 我说：“I never dream about it. I said I have nothing to do with it anymore.” 这就是整个故事，就是说这故事挺可惜的，但是我希望大家就是说听了我今天，我其实这个故事一开始是讲这个我做科研的理念不一定对了，因为这个是可能是有一点反潮流。我个人的这个科研就是一个，我是不愿意扎堆的一个人，所以包括我选择学校，选择就是住的地方，都是选择就比较 quiet 的地方，我不喜欢扎堆。要不是咱们这个 CADA 的这个领导们重视，我可能也不会在这里给大家分享。我很少，
没有没有，
我很少去
非常
这些会议，
嗯，好的。那施老师，那我简单介绍一下，然后我们就可以开始了啊。然后那
OK，
个，All right. So it's my great honor to host today's talk, Dr. Yu Guang, Roger Shi talk. So first of all, I would like to give a brief introduction of Dr. Roger. Actually, I know Roger for a long time when I was a graduate student. That was 2000 and I think it's six, something like that. when I was a graduate student, and because Roger is a good friend of my PhD mentor, Dr. Han Xiao. I think his training also in this audience. We had a lot of talk discussion about science, and also a good time with our lab member to have dinner together. That was I think it was about 16, 17 years ago, and when I was just starting my study. So just a brief introduction of Dr. Roger. Right now he is a distinguished chair professor at Bishop Aging Institute of University of Texas Health Science Center at San Antonio. Before he joined San Antonio, he was also working at Penn State University, and I think for about 10 years, I think. And during his work at Penn State, he identified SADAK kinase. I think right now, people in the lab, in Dr. Han Xiao's lab, also still working on that kinase. As Dr. Roger talked, he was originally working at Eli Lilly, right? He cloned PERK kinase. And later on, the PERK was win the Nobel Prize. And he also identified several lipid metabolism enzymes, including ALCAT, ALCAT, and also other kinase. Today, his talk will be mainly focusing on cardiolipin remodeling, which is leading to a major discovery of a first-in-class drug, so-called. Well, its name is DAFA, yeah. And DAFA actually, last time I think in Changsha I heard the Dr. Shi's talk, Dr. Rogers talk about DAFA is actually very powerful in treating diabetes and associated complications. So I think today Dr. Roger will talk about their major discovery about this new drug. And I would like to also thank Dr. Jiankang, right?
Jiankang Liu, yeah.
Is he here or not? Dr. Jiankang, Liu Jiankang?
Maybe he's busy.
He's accept the invitation to be a panelist, and he will also come to have the discussion with us about Dr. Rogers' discovery about the new role of cardiolipin remodeling in the new drug discovery and also treatment for diabetes and associated complications. So without further ado, let's welcome Dr. Roger for the talk. Thank you.
All right, thank you, Zhuo Xian, for the introduction. I'm very honored to share our work. I'm an outlier in scientific research because we don't do a very popular research to date. The reason is, in this preface, I want to share my philosophy of doing research. So research, in my opinion, is three stage of research. And the first is the amateur level, and the second is the professional, and third is the great master. Amateur is basically following impact factor. Wherever there's a hot topic, you just go on. And somebody catch a fish, you just follow him and find a hot spot. The professional can make a good living because they work on the same pathway for years after years, called 专家. Then the great master is draining the swamp or draining the lake and let the amateurs finish selling the fish for them. And when the amateurs finish selling each individual fish, the great master get a 炸油包奖, whatever, Alaska. Okay, that's my philosophy. Is that if you want to do something that eventually somebody remember, you have to drain the swamp. And unfortunately, nowadays, science has become a beauty selection. So seeing as change our career path, if it is a microbiota very hard, everybody jump on. Brownie is very hard, and everybody jump on. MicroRNA and so on and so forth. But unfortunately, If you do beauty selection, yes, you can get very good impact factors publications, but forever you will be amateur because you basically serving become a 韭菜. The great master is 韭菜. To year after year, you help them to finish all the pathway discovered by the great master, and the credit is belong to the great master. So we also did a beauty selection, but our beauty selection criteria is very different. The first is has to be very important question. The second is the groundbreaking. Third one is our work has to impact drug development, so it has to be druggable. Over the years, we've been working on 5 drug target identified in my lab. And of course, PERC was the first one become the international level beauty. That's because the big lab changed the name and adopted my child. We call the pancreatic alpha kinase, and it was changed to PKR like associated kinase. So because it's big lab, it immediately become a beautiful field everybody follow. But today I'm going to talk about, one of the drug target we clone. Oh, by the way, all the 5 enzymes, in my opinion, 2 of enzyme actually has generated, the groundbreaking part for new field. So of course, the PERK kinase stress and ALCAT actually together with LPGAT is the phospholipid remodeling, which is a completely new field before we contributed this development of new field. Before our work is very little is known. So this is ugly duckling, and every time when we submit to Cell Nature Science, the editor will say, well, lack of innovative conceptual advancement or innovation. I said, what? Anybody else work on this very important question? So that's how we've been, laying low, and so far nobody know about this very important work. So hopefully by the end of the seminar, you realize this is far more important work than the cloning and characterization of PERK. Because when Peter Walter was visiting us in 2019, I talked about this story. He take out his iPhone, start recording our conversation, because he was working for Calico. And I said, Peter, if you want my story unpublished one, we can sign the CDA because you represent a company. So here you have somebody who is the recipient of the Alaska Award and start documenting our unpublished story of ALCAT1. I hope this will be eye-opening for our audience today. So the title is In Search of the Holy Grail. Holy Grail, everybody know what I mean, but it's basically addressing a very important question in aging and metabolism. So we know that the world population is aging rapidly. So the upper panel is in China, lower panel in the US. So actually, my age is already getting to so-called old age. So aging actually itself eventually send us to another world, mainly because aging promotes the development of the whole family of age-related diseases. The most important disease is the cardiovascular disease and diabetes and neurodegenerative disease. Of course, cancer is also a big deal, but the cancer is more complicated. It's both aging and a genetic mutation, and so so those diseases has a very common feature is that they come in a group. So if a person has diabetes, that means this person must have other issues like hypertension, hypertriglyceridemia, chronic inflammation. So it's basically one disease manifested in different tissues. So if you just look at this common feature of this disease, they all happen in the tissue that are heavily dependent on mitochondrial function, which is the brain, the heart, the eyelid, the beta cell. Okay, those 3 tissues are heavily dependent on mitochondria for its basic function. Obviously, if let's say we have a hypoxia for 3 minutes, we will have a brain dead. We will have brain dead, and our heart will be stop working. Islet beta cell will no longer secrete insulin. So aging is everybody's nightmare. Eventually, we're going to lead the world. And so from very early on, the Chinese emperor is the leading scientist in study aging. He sent his primary physician Xu Fu to find a longevity drug. And Xu Fu is very smart. He never came back because there is no such thing. So obviously, if we study aging, we must pay attention to mitochondria. So the mitochondria aging theory was proposed a long time ago. It was by Daniel Hammond. He proposed that mitochondria control the aging clock. Mitochondria is like an engine of our automobile. It has a limited lifespan. You know, let's say you have a Mercedes-Benz, you could drive, let's say, 500,000 miles before it's totally blow up. And if you have a Hyundai, maybe 150,000. So the odometer is the aging clock. So here, mitochondria aging clock is measured by oxidative stress damage to mitochondrial DNA, phospholipid, and protein. One of the phospholipid is the cardiolipin, which is a very complicated phospholipid, and very few people pay attention to. That's what we fall in love with 20 years ago. But anyway, this cumulative damage, set a stage for the clock to, at the middle age, we are very sensitive to develop a whole family of age-related diseases. So why so? Well, more than 30 years ago, this is a work by Douglas Wallace. Actually, Wallace is one of the persons that are very adamant about, we're paying too much attention to the big genome and we pay very little attention to mitochondrial genome, mainly because the genomic era has already passed and we have not found a single gene that responsible for major chronic diseases. If any of the audience know a single gene that causes a common disease, let me know. None. As far as I know, is none. But anyway, so early work showed that mitochondrial DNA mutation is very adamant in the old population. Can people see the cursor? I'm moving around. Do people see the cursor? Yeah? Anybody?
No.
Anyway, yeah, if you cannot see the cursor, then my talk will become kind of difficult to follow. So here's the band. If you cut with a restriction enzyme, if there's no, this is a purified mitochondrial DNA. If there's no mutation, you will cut it. It becomes a single band. If it has a mutation, you cannot cut the DNA, it becomes a smear. So by middle age, most of our mitochondrial DNA has been mutated. So mitochondria is a fascinating organelle. It has its own genome, and it's population genetics. Each cell has multiple mitochondria. Like a liver cell, you would have more than 2,000 mitochondria in each cell. And each cell would have 5-6 copies of mitochondrial DNA genome. So this mutation reaches threshold by middle age. That's like somebody cut 80% of your liver. Now your liver is really in trouble. And that's at the threshold depends on what tissue your parents pass on, what kind of quality of tissue your parents pass on. That's where the genetic background play a role. So somebody could inherit a very strong heart and will not have developed heart disease, but could have a weak pancreas or pancreatic islet. That's where you develop diabetes. So even though age-related diseases is caused by the mitochondria, but it's manufactured in different tissue, different person. That's the fascinating part of aging research. Why mitochondrial DNA mutation not cause systematic problem in brain, heart, islet, beta cell? No, different people manifest in different tissues. So why study cardiolipin? Well, this is a phospholipid. When I was still at Eli Lilly, we were fascinated by cardiolipin because very little know about this phospholipid. Can people see the slide? Yeah. Okay. So this is the structure of a cardiolipin. This mitochondrial signature phospholipid. It's only found in mitochondria, nowhere else. It is required for every known mitochondrial function, from fusion, fission, mitophagy, mitochondrial DNA biogenesis, inflammation, everything. And just like any lipid, the function of the cardiolipin is determined by the fatty acid composition. In healthy mitochondria, in healthy tissues, when we are young, in the metabolic tissue, the four fatty acid chains are dominated by linoleic acid. So we have expert in lipidomics. His name is Xianlin Han. I think he's probably in the audience. Anyway, Xianlin's work demonstrated that during onset of diabetes and later on by other people study other chronic diseases like heart failure and stroke, that during onset of age-related diseases, there's a gradual replacement of this 18 carbon 2 double bond fatty acid chains with very long polyunsaturated fatty acid like DHA, arachidonic acid. This change is so powerful that the whole mitochondria, every part of mitochondria is in trouble. From fusion, fission, mitophagy, and mitochondrial biogenesis, inflammation, and then the chronic disease. So here's Xianlin's very early work. Xianlin and I actually collaborated a long time, and finally I decided to convince our school to recruit Xianlin about 5 years ago. He's a rising star here. So his early work demonstrated that during onset of diabetes, you have a depletion of the good cardiolipin, which is a tetralinoleal cardiolipin, the 18 carbon 2 double bond. There's the enrichment of the DHA cardiolipin. And later on, other people are also finding other age-related diseases like heart failure. You have a depletion of the good cardiolipin and the enrichment of the bad cardiolipin. So my lab is fortunate that we pioneered the early work in the mitochondrial cardiolipin metabolic pathway. We cloned several pivotal enzymes involved in cardiolipin synthesis and remodeling. The synthesis is the cardiolipin synthesis gene, which I don't have time to talk about today. And the remodeling enzyme, there are 3 of them, which involve in phosphatidylglycerol remodeling and cardiolipin remodeling. So today I'm going to focus on this cardiolipin remodeling enzyme. So why need the remodeling? Well, because cardiolipin is localized in the inner mitochondrial membrane, it's constantly bombarded with free radical. The free radical damage would cause apoptosis if not repaired. Actually, the last step of apoptosis, which is demonstrated by Sheldon Wang's work a long time ago, is a dissociation of cardiolipin from cytochrome C. That's where it triggers mitochondria-mediated apoptosis. So oxidized cardiolipin must be repaired. The repair process is faster than resynthesis. It's like your car. Once your car breaks down, what do? You quickly find a spare tire or you find whatever spare parts and replace it. So that's what happened to mitochondria cardiolipin. So oxidized cardiolipin must be repaired. The repair process takes 2 steps. The first step is the hydrolysis with this enzyme, cytoplasmic phospholipase A2. And so now you have a lysocardiolipin, which is a lack of one acyl chain, then followed by acylation. So this is really hardcore lipid biochemistry. And each time when we send a paper to the editor, they just don't understand the whole process. And they quickly return to us, the lack of innovation, lack of a conceptual advancement. I say, okay, you're a genius. And we will, and most of the time, we don't even bother to submit to CNS. Not because they're not good journal, but they the beauty selection. They do beauty selection, and ours is ugly duckling. Anyway, so the second step is where you determine the final outcome of the fatty acid composition of a cardiolipin. So there are physiological remodeling enzyme, which is carried out by tafazin. Tafazin, when mutated, will cause premature death due to the a rare genetic disease called Bar syndrome. And those kids die very young because of heart failure. Remember, heart failure is old man's problem. And so this is a very good enzyme. It will recover the original healthy structure, which is a tetralinoleal cardiolipin. Then the pathological remodeling enzyme is the enzyme we cloned. We're very lucky because this enzyme is not druggable. Tafazin, you cannot activate for tafazin to treat any disease other than using gene therapy. Now, ALCAT actually does not choose the linoleic acid CoA as the donor. It chooses DHA and other very long polyunsaturated CoA. And after remodeling, produce a very bad looking cardiolipin. Now you have a cardiolipin no longer symmetrical. You have one chain very, very long. That's where you find in all aging, all tissues. So this enrichment really mimic the aging process. It took us almost 10 years to find out this is the bad guy. After we studied so many basic biology of this enzyme, including the phenotype of knockout mice. So here early on, we realized that if you generate a stable cell line, most of the cell will die. And few cells that survive, they have a depletion of a tetralinoleal cardiolipin, which is a good cardiolipin. And they have an enrichment of bad cardiolipin. When you ask me why there's so little bad cardiolipin, the reason is they're very unstable. They have so many double bonds, they quickly oxidize and then being depleted. Okay. So this actually is the beginning of my collaboration with Xianlin. Actually, Xianlin charged me arm, legs for this figure. I still feel disgruntled about this. It's a very expensive figure. So we made a knockout, and sure enough, if you knock out this enzyme, this is in diet-induced obese mouse, the good cardiolipin go up. Okay, this is a tetralinoleal cardiolipin, and this individual bar graph represent a different cardiolipin species. You can read our paper published long time ago. So that really set the tone about the function of this enzyme. So let me go through some basic stuff before I get you lost, because well, you talk about something that are hardly understandable to general public because the lipid metabolism is not a fashionable field. So why cardiolipin is so important? Well, cardiolipin play a pivotal role in mitochondrial structure. Mitochondria has two membranes, the outer membrane and the inner membrane. The inner membrane is protein rich. It is actually most protein rich place in our cell. That's because the mitochondrial inner membrane has compact with curvatures called a cristae. Due to cristae, the inner mitochondrial membrane surface has enlarged more than probably 10,000 fold. It's very difficult to calculate. You have so many curvatures. That curvature allows the mitochondria oxidative phosphorylation complex to be crowded together. This curvature requires cardiolipin because cardiolipin has this cone-shaped. Without cardiolipin, you will not be able to make the curvature. So when cardiolipin is depleted, for example, if we overexpress our enzyme, the inner membrane becomes flattened. Now the five complexes become far apart. The oxidative phosphorylation complexes fall apart. Now you produce very little ATP, you generate a lot of free radical. That's the mechanism of action. And each time when we talk to the editor, they just don't get it. And they say, where is the mechanism of action? So here is another proof that if this is our stable cell line, this is vector control. You can see due to the invagination of curvature, the vector control mitochondria is very black, very dark. So if you overexpress ALCAT, the inner membrane be flattened out, the mitochondria becomes swollen because they cannot go through the fusion fission process. So let me go through again the major difference from linoleic acid, which has 2 double bonds, and the DHA, which has a 6 double bond. And other people published work early on showing that the DHA content in cardiolipin is proportionally correlated with the peroxidation index, which means that the lipid become easily oxidized. Oxidized lipid will generate lipid peroxide, which is a very bad form of reactive oxygen species. So that's how the correlation is very close correlation. The more double bond, you have a more oxidative stress in your cell and production of free radical. So we demonstrate this in the stable cell line. If we overexpress this enzyme, you can see there's a very high level of oxidative stress. Sky high. And due to this oxidative stress, it causes mitochondrial DNA mutation and cytoplasmic release of mitochondrial DNA. So this is a percentage of mitochondria being released in the cytosol if we overexpress our enzyme, okay? And this release causes activation of the STING pathway, which is the contribution, well, discovered by James Chen. I think James Chen already received the Breakthrough Award. We wish him good luck, and the next step it will be the last guy. So with that, we try to answer several questions. The first question is obvious one. Does ALCAT control lifespan? Because this is the holy grail that people has been studied for decades and hundreds of years, what control our lifespan. So here's another circumstantial evidence. This is published a long time ago. And the DHA content in cardiolipin is inversely correlated with longevity. That means the higher content of DHA, the shorter of a species lifespan. Like a mouse has very high content of DHA in cardiolipin. So they only live a 3.5-year-old. Horse has a very low and a human somewhere here. And that's really support that this enzyme probably promotes aging. Another amazing discovery or phenomenon was the lifespan of a clam. We view clam as our food, seafood. But actually, clam has two types of clam live a very different lifespan. The black ones live 500 years old. Actually, about 6 months ago in Boston, somebody found a clam that was over 200 years old. That clam was still alive, was just already born when Washington still in power. And that's how old this clam can become, a 500-year-old. And there is a sibling clam, only live 50-year-old. Some people study aging, ask a question, what's the difference between the two clam? Well, it turned out that still the DHA content. If you have a very low DHA content, you could live as a clam, you could live 500-year-old. If you have a very high DHA content, you only live 50-year-old. And that is correlated with the peroxidation index as well. The longer the clam live, that's the peroxidation, like in human, our lipid peroxidation index is very low. That's why we live very long life. And this is the liver mitochondria peroxidation index. Once again, if you have a very little DHA cardiolipin, you will have very little peroxidized lipid, and you live very long life. And from clam to human. So we use several aging models to study how ALCAT control aging. The first is the natural aging. And, our mice live not forever. They only live 15, maybe 20% longer, not big deal. But in mammal, you could triple your lifespan in C. elegans, Drosophila, but the higher mammals, it's very difficult. This is a natural aging. Our mice actually live a 3.5 year long. Compared with, most published data, our mice live very long. And so by knockout ALCAT, they live 10 to 15, probably 15, less than 20% longer. The most remarkable thing is they have a healthy aging. So this is a typical 3.5-year-old mice. They have a gray fur, they have a hunchback, and they are sluggish. They don't move. They sit in a cage. And if you walk into the mouse room, you can tell the difference between this cage and this cage. Our knockout mice just like a young person, very active, have a shining fur. They don't have a hunchback. And they have a very good cardiac output. This is the echo study of cardiac output. Their heart like young mice, in the ejection fraction shortening, and they have a very good glucose homeostasis. This is a glucose tolerance test. Our knockout mice, barely have a hyperglycemia when you challenge them with glucose. They have a very strong muscle strength. This is the rotor test. This is volume grip test. That means their muscle is much more powerful than the control mice.
哎，好像
Want to
有点
summarize?
卡
This
住，
is like I want to summarize most. Yeah. What?
刚才
Can
刚
can
才
you hear me?
刚才有点卡住了，我不知道是不是我的问题。It's
Okay.
okay right. It's okay right now. It's okay right now. Yeah.
Oh, okay. I'm using my home Wi-Fi. I think it's probably very strong. Okay. So
Yeah.
the second model is mitochondrial aging mice. As I begin with, I bring you the early work of Doug Wallace. By the way, Doug Wallace also been nominated quite a few times to receive the the Nobel, but I think he's still need more proof, like our work. So the second model is the mitochondrial aging mice. So Doug's very early work is he's generated mitochondria DNA mutation mice. They call the POG mice. Those mice has a premature aging. They have by a year and a half they die of a heart attack. They have loss of a muscle mass. They have a gray fur. They have a hunchback. So we cross our knockout mice with the POG transgenic mice. The POG mice live a year and a half. They die. And if we knock out our ALCAT enzyme, they live much, much longer. This is the male mice. This is the female mice. Okay. And more importantly, you cannot miss them. They don't have gray fur, they don't have hunchback, and everything you measure, they look like young mice. And their cardiac output, as I told you, that POG mice die of heart attack. They die a year and a half. Because just like old man, most of us would die. Our premier just died from a heart attack. That's, he was still a pretty young guy. Anyway, so the ejection fraction, the POG mice, by a year and a half, they lost 30% of their ejection fraction. This is a measurement of cardiac output. If we knock out our enzyme, it completely normalize the cardiac function. This is the measure by ejection fraction shortening. You can see other people publish their mouse model. This is the same POG mice. See how ugly they look. But our people are not very good in taking good pictures. So because mitochondrial DNA mutation will cause release of mitochondrial DNA from inside mitochondria to the cytoplasm, as I showed you very early on that this process is controlled by the ALCAT. So sure enough, this is the POG mice. This is measured by different mitochondrial DNA loop by PCR analysis. So you have a very high level cytoplasmic DNA content. This is a different region of the mitochondrial DNA. If you knock out our enzyme from the POG mice, you completely prevent this cytoplasmic release. And that will completely prevent the STING pathway activation. This is what happens if you knock out ALCAT. This is the quantification. So mitochondrial morphology also have a dramatic difference. So the POG mice is mitochondria fragmented because the mitochondrial DNA mutation causes the whole mitochondria behave very weirdly. They have fragmentation, and they have mitochondrial DNA copy number also go down. If you they also have other defect, which is not indicated here. So if you knock out ALCAT, you can see the POG mice is packed with mitochondria. Their crystal structure is very, very clear. Here you can see the crystal structure start, getting smeared, and also restore the mitochondrial DNA copy number. So naturally, this oxidative stress caused by mitochondrial DNA mutation would cause the telomere shortening, and telomere shortening is a very good hallmark of aging. That's how Elizabeth Blackburn received a Nobel Prize for the discovery of a telomere. So we use the telomere shortening mice to prove that indeed ALCAT control the process. Once again, we cross the ALCAT knockout mice with telomere shortening mice. You have to back cross fourth generation. By fourth generation, because the mouse has very long telomere. You can see that by knockout our ALCAT, you will not restore the telomere length because the telomere guide RNA is no longer there. You cannot produce telomerase anymore. Okay, and then both in male and female they have telomere shortening, but they do not develop age-related diseases. So this is morphology-wise. You can see the edge-turret mice, knockout mice, they have a hunchback. All right, those mice didn't show much periphery yet, but our knockout mice doesn't have this. The generation, fourth generation, the single knockout, the edge-turret knockout no longer fertile, so you cannot pass on more than fourth generation. This is a very expensive project. Actually, it took my graduate student John Paul 2.5 years to generate phenotype, and he was very upset. And by the end of it, by the time he's graduating, all the data being generated. So those has yet to be published. We're going to publish very soon. So you can see it's remarkable by third generation. By knockout, our enzyme doubled the lifespan of the edge-turret mice in male and female. More importantly, they have normalized the morphology or phenotype from body weight to oxygen consumption to cardiac function. So you can see by third generation, the edge-turret mice have a cardiac problem. And by knockout, our enzyme will restore the cardiac function, both in male and female mice. I'm going through very quick because this is very late in China. I want to make sure you go through the data, but we will have time to ask questions. So menopause is the milestone of aging, and so is telomere shortening. Telomere shortening by fourth generation, the edge-turret mice no longer fertile. They stop producing young. This is our double knockout. You can see they're still producing young. That's because they have a normal morphology of the testis. So this is a wild type testis. This is edge-turret testis, and this is a double knockout. You can see mitochondrial morphology play a role in projectile of a sperm. The sperm has to swim and find the egg. So mitochondria play a big role in that process. So the tail of a sperm is wrapped in with mitochondria. You can see this is wild type mice. This is our double knockout mice. They're very similar. This is the edge-turret knockout mice. The mitochondria is gone, okay? Pretty much gone. And there's no sperm. And you will naturally ask, well, how big a deal with the cardiolipin? Well, very important. So by third generation, the edge-turret mice has depletion of tetralanormal cardiolipin. This is a knockout of our enzyme, restore most of the tetralanormal cardiolipin, which is a good cardiolipin. And this is a bad cardiolipin go up, and this is after knockout enzyme, it depletes the bad cardiolipin. This is I believe a male and a female mice. Both are very similar. So with that question, we want to ask the final question, which is cellular aging. Because cellular aging is called a senescence. It becomes very fashionable after 20, 30 years, discovery of senescence. Now, all of a sudden, the big lab has recooked it and say, well, cellular senescence is a big thing. It controls every part of aging. Then let me test it, okay? So cellular senescence actually is protective from development of cancer. So that's a big thing. So we live longer because of cellular senescence. When we are young, our cellular senescence prevents the development of cancer. But when we get old, we have to pay price. The accumulation of senescent cells promote cancer development, promote the development of age-related metabolic disorders. So aging promotes cellular senescence. Telomere shortening promotes cellular senescence. Mitochondrial dysfunction promote the cellular senescence. So I already talked about the three models. Okay, now let's look at the cellular senescence. Well, this is where it brings another renaissance for cellular senescence research. This Nature paper basically using a transgenic mice to deplete the cellular senescence. When I look at this, I say, very interesting. And there's a company invested billions of dollars targeting cellular senescence still going on. But I was very skeptical, mainly because you cannot generate a transgenic human to deplete the synaptic cells. But you can use our enzyme inhibitor to do so, mainly because during cellular senescence, our enzyme is upregulated. So this is isolated mouse embryonic fibroblast, MAF cells. So you can culture them in vitro for 5 generations, they become synaptic, no longer grow, no longer double. And very early on, we found that if we knock out our enzyme, the MAF cell can continue to grow another 10 more generations. Next passage, our enzyme already upregulated. And this is a knockout. So you can see that this is due to the upregulation of synaptic biomarker, which is P53, P21, P16, and others. If we knock out our enzyme, those synaptic biomarkers all normalize, okay? And most importantly is the mitochondrial morphology and function. So at passage 2, they were very small. The synaptic cell become really enlarged by passage 10. This is the MAF cells in the wild type control. This is our knockout mice MAF cells. You can see they remain small. That's because basically synaptic cells are sitting there doing nothing. We're just consuming energy and not contributing anything. If you look at the mitochondrial morphology, by the time passage 6, the mitochondria become swollen. They no longer go through the fusion, fission, mitophagy cycle. And if we look at our knockout mice, MAF cell, they have a normal mitochondrial morphology. You can see they're very small, no longer, those are very gigantic mitochondria. Even passage 10, they still look normal. If you look at using seahorse, you measure mitochondrial respiration, which is a gold marker for mitochondrial health. And at the low passage, there's no difference. Passage 6, the control cell already dropped by 30%. By passage 10, our knockout also start losing the steam and the respiration also dwindles. So in collaboration with G.D. Campisi, who is really a guru in the cellular senescence research in aging, we get the reporter mice, which is the P16 gene promoter driven the expression of EGFP. So you can image the live mice to see because P16 positive cells are synaptic cells. So you can induce cellular senescence by feed mice with high-fat diet. So it makes sense because high-fat diet, obesity promotes obesity. Obese person have the phenotype of old person, like heart problem, diabetes. Those are old man's disease. But if you get obese, you basically accelerate the aging. So this is the wild type mice. You can see the abdominal fat, there's a lot of synaptic cell accumulation. If we knock out our enzyme, they all gone. But you could argue, well, because your mice is skinny. Well, I'm going to show you with the inhibitor because we cannot do the inducible knockout in adult mice, but we can use the inhibitor. I'm going to show you that doing the same thing. If you knock out the ALCAT, there's no accumulation of synaptic cells. So with that, we want to see the connection between aging and aging-related diseases. This slide alone cost us more than 10-year work, and some part of it still remains to be published. So we show that if we knock out the ALCAT, the mice are completely protected from every age-related metabolic disorders, from obesity, type 2 diabetes, fatty liver, renal failure, diabetic cardiomyopathy, fibrosis. And this is the mitochondria. In the obese mice in the heart, there's a lot of dead mitochondria. This is a knockout mice. This is the renal failure mice. from STZ-induced diabetic mice. They have a problem with the renal globular structure. You can see the loss of the the thickening of basal membrane. This is the knockout mice. You can see they have a normal looking. Browning, another thing that I always laugh at. I personally don't believe browning play a big role, but this is a fashionable part, so we did it. If we knock out this enzyme, this is tissue-specific knockout, you prevent the widening of the brown fat. This is the high fat induced brown widening of the brown fat. If we knock out the ALCAT, you prevent that. Oh, by the way, you can see that the ALCAT is induced by high fat diet. And in doing so, they restore mitochondrial DNA copy number. So diet-induced obesity deplete mitochondrial DNA copy number in the brown fat. And by knockout this enzyme, also restore the expression of UCP1 because high-fat diet will cause a depletion of UCP1. You can see this is UCP1, it's gone. If we knock out the ALCAT, it come back. So remarkably, we also did in vitro browning induction stromal vascular cell. After 6 days of induction, you can see the lipid droplet accumulate. This is typical white adipocyte. This is our knockout white adipocyte. It's amazing. It's basically look like a brown adipocyte. It has a lot of mitochondria. The lipid droplet is very tiny. So another very difficult disease, which I hope all of us, none of us would ever have it, is AD. AD is basically a dead man walking. You have no memory. And so we use the transgenic overexpress A beta and PS enzyme. They develop a AD-like symptom. They have accumulation of A beta plaque. This is our knockout mice. Most of the plaque is gone. The behavioral tests show they basically have a normal memory. This is the water maze test. This is the AD mice. This is our knockout mice and double knockout mice. To do with religion, we use this as slang. So the legends say that this cup has got lost. The loss of the cup. If we can find the cup, this is have a mythical cure power for every disease. So I mentioned that aging is the driving force of whole family age-related diseases, mainly manifested in mitochondria-dependent tissue. They are one disease manifested different tissue. Currently, the treatment is symptom-based, very primitive to say the least, because AD has no treatment. Heart failure, if we have a treatment, our premier Li Keqiang probably is still alive. And so is a stroke. Very few people, other than, within 3 hours, you get, the blood vessel being really, the clot being melted and or get a stent. Other than that, there's no really effective treatment. So the holy grail is that you have done those are in rodents. Who cares? We cured every disease, including cancer, many times in rodents. So who cares? You cure this once more. So I'm going to show you that the most powerful part of our work is develop a drug that eventually can benefit every one of us. So the question is, if we have a ALCAT inhibitor, can we target all age-related diseases as one disease? That's what I try to answer. So this slide cost us about $22 million so far. So because my first career is at Eli Lilly, that's where I led the development of the first GLP-1 analog, which is not big deal because I happen to be the lucky guy, the lucky place, but it takes about 25 years now, have a semaglutide and tirzepatide become the magic weight loss drugs, which I'm very glad to see that. But those weight loss is not going to solve the problem of aging, okay? So we spent 9 years for the target identification validation. We did a high throughput screening when I was still at the Penn State. We did a structure activity relationship study by synthesizing more than 1,000 compounds that improve the potency of the inhibitor. And we also did a lead optimization by identifying clinical candidate. Now we have 3 clinical candidates. One of the clinical candidate is dapagliflozin, which is we did all the proof of concept study in my lab. So DAPA is very potent. This is the enzyme product, not the enzyme expression level. This is the enzyme product, which is the cardiolipin itself. You can see we can add very little, like 10 nanomoles of dapagliflozin is still inhibit about 50% of the enzyme activity. The cross-reactivity is 0. So this is 100 micromole inhibitor. This is closely related to enzyme ALCAT1, ALCAT. They have 0 inhibition. With ALCAT3 as maybe because this is very high, 100 micromole versus 10 nanomoles. So by 33 micromole, it does nothing. So this drug is as clean as you can get because we spent 8 years. Well, not when I say 8, we is the part of my lab and the company that we co-founded. The inhibitor is 7 nanomole IC50, which is as good as you can get as the enzyme inhibitor. So the first disorder we try to treat is the cardiovascular disease. We use the heart failure model. My graduate student used the ligation of a descending anterior coronary artery. Those mice will develop typical heart attack. They will die within 24 hours without treatment. Or if we treat it with a vehicle or with a DAPA. With DAPA treated, there's no death. And the ligation of the coronary artery is like somebody had heart attack. That's what a premier Li Keqiang had. So I don't know whether he had a coronary artery blockade or whatever, but they die very quickly because the left ventricle function is lost. If you treat with our inhibitor, which is a 10 nanomole, 10 milligram per kg, that's the daily dose we use twice daily by oral gavage. You can see we restore the left ventricle function. This is the echocardiograph. And it will prevent a dilated cardiomyopathy. This is a dilated cardiomyopathy. This is the vehicle treated. This is DAPA treated. And there's a hypertrophy of the vehicle treated of a cardiomyocyte. This is DAPA treated. So you can see this drug is really a miracle. I mean, here you have mice which have a total ligation of the descending coronary artery. It will partially prevent fibrosis because after all, the tissue lack of oxygen, part of the heart, but they will develop a certain level of fibrosis, but it's much less than the vehicle treated. The mitochondria is also the morphology being restored. This is the heart failure mice, their cardiac mitochondria is basically fragmented. You can see they, due to the fission defect, which we already published part of it in the PNS paper. And if we treat it with a DAPA, you see mitochondria have a normal morphology. And not only that, the DAPA really mitigate lipid peroxidation. This is the measurement of lipid peroxidation, the 4-HNE. This is vehicle treated, this is DAPA treated. This is remarkable. The oxidative stress is completely gone. And also restore the tetralinoleal cardiolipin in the heart. This is the heart failure model. This is treated one. And depleted bad cardiolipin, which is DHA. And we use another model, which is the constriction of the aortic artery. So this is called a transaortic constriction. This mimics a person who has a hypertension, a blood vessel blockade, and this kind of, hypertension, hypercholesterolemia patient. So those mice also develop dilated cardiomyopathy, and the treatment also prevented. You can see this is morphology. This is a left ventricle function measurement. This is fraction shortening, fraction ejection fraction. Each cases, you can see the cardiac function is restored. And likewise, mitochondrial fragmentation is completely gone. This is, by the way, those mice were treated 4 weeks after the constriction. So they are still low level mitochondrial fragmentation compared with, the heart failure model we treated same day. But nevertheless, from here to here is remarkable improvement. The fibrosis is completely gone. So another very difficult to treat disease is diabetic complication. You could have a semaglutide. You could have all the cholesterol lowering drug, but diabetic complication will increase as you get older. This is published a long time ago. New England Journal of Medicine, you can have a perfect, you have a normal glycemia. That means you can control your blood glucose to a lower level. But if you follow those patients, by the time they're getting older, they still have a cardiac problem. They're getting worse and worse each year, and the renal problem, retinal problem. So retinopathy is really one that kills diabetic patients. So we use the STZ mice to, as a model for diabetic complication. We give them STZ, then 2 weeks after onset of diabetes, we give them DAFA in the supplement. We supplement DAFA in the food. So we measure 4 months later, we measure the cardiac function. This is so actually the heart become heavier when they develop diabetic cardiomyopathy. This is the vehicle control. This is the DAFA treated. This is their cardiac function. This is the vehicle treated. This is DAFA treated. Completely restore the cardiac function in diabetic mice. Fibrosis equally being mitigated. This is the DAFA treated. This is the STZ vehicle treated. Mitochondrial fragmentation, once again, being completely mitigated by DAFA treatment. And another amazing thing is that those mice are diabetic. The STZ has destroyed their islet beta cell. But some of the mice actually, their blood glucose try to remain normal because this is the STZ diabetic mice. All of them develop hypoglycemia. This is after 4 months treatment. Some of our mice actually maintain fasting blood glucose between diabetic and non-diabetic. This is a glucose tolerance test that also indicates that. So that means DAFA actually restored some of the islet beta cell function, which my lab actually stopped working on the islet beta cell, mainly because for years we could not get funding. So which is turned out to be a very good thing for us to focus on the ALCAT story. So another complication is diabetic renal failure. So this is diabetic mice. They urinate a lot. This is urine volume in the STZ vehicle treated. This is DAFA treated. They have no, they don't urinate more than the control mice. And equally so, they don't have, if you measure the protein in the urine, they have very little protein in the urine. So it's consistent with improvement of diabetic renal failure. And another golden marker for diabetic renal failure is the hyperphosphorylation of kinase. You can see this is STZ vehicle treated. This is DAFA treated. It's completely normalized. So finally, well, the third model we use is the obesity and metabolic complication, because obesity is a major issue in both in China and the US. We are not trying to develop another anti-obesity drug. There's nobody is going to develop a drug better than tirzepatide and semaglutide. The third generation is just the wonder drug. But this work was done quite a while ago. To show you that the DAFA does improve obesity. It and as I mentioned, here is obese mice. We use the cellular senescence mouse model. You can see obese mice has a lot of senescent cell in the abdomen. Here we give obese mice DAFA. It does not reduce the body weight as much because this is given after 3 months of obesity. So it takes time to lose weight. But synaptic cells almost completely depleted by DAFA. So DAFA actually is the first non-BCL2 inhibitor-like senolytics, because currently all the senolytics are targeting BCL2, which is the apoptosis protein. Likewise, DAFA also mitigates obesity-related metabolic complications. This is the fatty liver model. DAFA completely mitigated fatty liver. And DAFA restore the white adipocyte beiging ability. So this is vehicle control. This is white adipose tissue lost its browning ability. This is pretty much a beige adipose tissue, because white adipose tissue under normal diet, they have a lot of beige fat. You can see the color is different. High-fat diet would induce the defective beiging abilities that become very widening. A lot of lipid droplets become swollen. And this is DAFA treated. So this is also a thermogenic compound. And amazingly, DAFA treatment upregulate UCP1 by sky high, and also TRDM16. This is a shingles antibody they provided us a few months ago. So those are amazing because it's better than the knockout mice. Finally, I'm going to talk about this most difficult to treat disease, which is AD. AD is frightening because when you get older, 65 and older, you start to have a dementia. You agglomerate, your memory is no longer that sharp, and eventually like Walking Dead. And so there's virtually no effective treatment. A beta antibody is still oversell. I don't think only small population show some improvement of a beta antibody, and it also causes brain hemorrhage, bleeding. Those things really are troublesome. I'm not sure it's going to be a blockbuster. So here's what we find out: ALCAT in AD. ALCAT expression in AD patients gradually increased as the symptom getting more severe. In the transgenic mice versus the non-transgenic, you can see the A-beta transgenic mice has sky-high ALCAT. So A-beta basically overexpressing induced ALCAT. So ALCAT is basically a mediator of AD phenotype. So using, previously I showed you the knockout mice. Here is the DAFA. DAFA, this behavior study is still quite difficult to do. I mean, the intergroup variation is very big. I mean, we don't have, my lab this is mainly done in China. Students are learning how to do a behavior study, but nevertheless, we repeated several times, and DAFA each time improved the memory of the AD mice. This is two different measurements. You can see significantly improve memory. And not only that, DAFA restored the morphology of the brain neuron in the different region. You can see this is the wild type neuron, this is the AD neuron, this is DAFA treated neuron. They are in between the wild type and the AD mice. You can see the individual neuron. More importantly, DAFA treatment completely mitigate the hyperactivation in microglia. This is microglia activation, make them very big and visible. This is a DAFA treated one. And this is a quantification of the inflammatory biomarker in the brain. This is IL-6, TNF alpha, IL-1 beta, and interferon gamma. So basically, to summarize that, I have shared with you the groundbreaking work we started 20 years. Begin with the bench work, the cloning, identification, the first enzyme involving phospholipid remodeling, to pathological studies of their involvement in aging and age-related diseases. So far we have tested, this is a dapagliflozin tested. In every case, it show a remarkable efficacy. So here comes this is the Alexander Fleming actually saved all of us. I think only maybe 1/10 of the population, 1/10 of the audience is still here, because most of us will die when we reach to 30, 35, 30, 36 age. In China, before I think before the discovery worldwide, because this is what it shows. Penicillin has doubled our lifespan within basically a few years. And now we reach a plateau because aging. Aging is a modern disease our ancestor never had. So hopefully our dapagliflozin will push up the healthy aging and further extend our lifespan. With that, I would like to thank my collaborators all over the world. The mitochondrial field is mainly David Chen, Shingo, Hiromi Sasaki, and senescence Judy Campisi, or mitochondria also Orion Shirahai, and lipid with Shenlin Han, and we collaborate with Dr. Han's lab in another Han in China, and the rest of the team. And here's the people who contributed to this study. I don't have time to introduce each one of them, but they actually are my disciples, and they really part of this adventure in the last 20 years. With that, I want to thank you, each of you, and I'll be happy to answer questions.
Okay, great. I think Dr. Rajesh for this wonderful talk. As you can see, it's a very long journey from the beginning. From the at very beginning, the cloning of the enzyme ALCAT, and so later on how they actually did a lot of work for the knockout tissue specific knockout using multiple models to prove that the genetic deficiency will have beneficial effect, mainly through mitochondrial cardiolipin remodeling. Then later on, amazingly, this I actually identify a new drug, DAFA, that is so powerful, can selectively inhibit the enzyme activity of ALCAT, and have been proved to be very efficient in treating many diseases. So now we are opening for the questions. Wendong, you have
have a question.
You have the question. Okay, right.
Thank you, Roger. Great talk. Yeah, DAPA is amazing. So have you done any preliminary study on the toxicity of the DAPA?
Yeah, we did that in our molecular metabolism, not molecular, it's called
Therapy.
In one of the molecular therapy, whatever, that journal, we published the phenotype of the heart failure model. And in the supplemental part, we published the chronicle toxicology study, because we have gone through a multiple round of discussion with a major pharmaceutical. Each time, the first question surfaced from the discussion is that, do you have toxicology study?
Right.
Yes, the company and the lab has made an extensive effort to make sure this compound is safe. So first of all, we did the toxicology study by CRO company in China, paid somewhere like $500,000 Chinese yuan to study in rodent model, mainly in rat. So they gave 10 times the efficacy dose for 2 weeks. Then they analyzed liver function, all the tissue morphology, whatever toxic marker related to toxicology, none. And this has been repeated by a major pharmaceutical. They have taken our compound, they did extensive study, including genotoxicity. The finding is that there's no toxicity. The third part is carried out by my lab in Nanjing Medical University, that we use once again 10 times the dose for entire month. We dosed 10 to 15. It's 100 milligram per kg. Efficacy dose is 10 milligram. For entire month, we didn't see any toxic. 9 months, there's virtually no indication of any toxicology.
Right.
Yeah. So we feel pretty safe. I'll be the first one to make a tea every morning, drink DAFA, because I need it. I'm getting old.
You want to live longevity, okay. But have they ever done the MTD, the maximum tolerance dose? I mean, the lethal dose.
Yeah, that's another very good question. Because our students have to publish their data and they graduate and find a job, so those work need to be done. We need to find a minimum efficacy dose versus a maximum tolerable dose.
Right.
So, this is very important for cancer. We have a graduate student just graduated last week, and we have 2 cancer studies. And once again, in the cancer study, it's very important to find the right dose, because the lower dose may not have efficacy. Very high dose could have a toxic effect. So the answer is no. We haven't done anything to titrate the what's the optimum dose for each disease.
Great.
Okay. All right.
Thank you.
Actually, maybe before maybe let Mei ask first. I can ask later. Yeah, Mei, can you do? Yeah. All right.
Go ahead.
Hi, Raj.
Hi, Lillian.
Yeah,
thank you.
Thank you very much. I learned a lot. Well, I have 2 small questions related to ALCAT biology. So looks like this enzyme really triggers aging process. So the first question is how this enzyme is regulated at transcriptional level and also post-translational level. I thought it's enzyme, whether they can be modified after translation, also transcription level. That's the first small question. Another question, you really touch on the multiple system and use different model. Did you really look at immune system for this protein? So that's my 2 small questions.
So Milian, I didn't catch your second question because you have a humming sound in your background. Can you repeat the second question? I'll answer the first one.
Oh, the second question is very simple. Did you get a chance to look at immune system of your knockout model?
Really?
The immune system.
Oh, immune system. Yeah, of course. Great question. NLRP3 activation requires oxidized cardiolipin. So cardiolipin is mainly residing in the mitochondrial membrane. Upon oxidative stress, it will promote externalization of oxidized cardiolipin to the outer mitochondrial membrane. That's where it serves as an assembly platform for the assembly of NLRP3 inflammasome. So this is not our work. It is published by big labs. So cardiolipin oxidation triggers activation NLRP3 inflammasome. NLRP3 inflammasome activation play a major role in chronic disease. From AD to diabetes to obesity. So what happens if you knock out ALCAT? It will prevent NLRP3 activation in obesity. That's how powerful it is because it prevents oxidation of cardiolipin. So answer your question. So modification of ALCAT play very important role in controlling its activity because as I begin my talk, telling you that we could not generate a stable cell line with a very high expression of ALCAT because that would kill the cell because of oxidative stress. So the cell actually survived the selection to become a stable cell line has a mediocre expression of ALCAT. I mean, those cells actually mimic senescence. They have a high level of oxidative stress. They have a enlarged morphology. The cell look very big and they spread just like senescent cell. And so that's why our body has to control this enzyme, make sure it does not really express much during normal time. Indeed, this is done by several levels. At the transcriptional level, it's induced directly by oxidative stress. And we have yet to find out which transcription factor upregulate, stress-induced transcription factor upregulate ALCAT expression. So because myself was trained in molecular biology, was very afraid of doing gene bashing, promoter bashing, so we didn't do that. Maybe someday, well, whoever in the audience could be happy to do that, and we can help. That's one level. The second level is the post-transcriptional level. So we just submitted a paper, and currently in review, this paper disclosed the phenomenon that ALCAT normally under normal condition, ALCAT is not expressing, mainly because lack of messenger RNA is there is a translation control mediated by the ER stress pathway. I shy away from ER stress pathway because it was just such a popular field. My philosophy is that once the field becomes very popular, I'm going to walk away. And same is ALCAT. If someday some big lab publish Nature Cell Science ahead of us, then I'm going to just change the field because we have a better field, another field to work on. But anyway, so we find out that there are two open reading frame upstream the real open reading frame of ALCAT gene in human. In rat mouse, there's additional open reading frame upstream the first ATG. This open reading frame serves as a decoy to prevent the translation initiation under normal condition. Only upon oxidative stress, it will promote the charged tRNA to find the real open reading frame, which basically is the same as the ER stress response, like RE1, all those ER stress responsive proteins. They have a 5' UTR, ORF, serve as a decoy that prevent the translation initiation. So if we delete the 5' off, it no longer regulated by oxidative stress. Okay, that's two level. So I believe you also asked the question why this enzyme is there if it is so bad. Did you ask that or you didn't?
No, that you answered all my questions. Thank you, Raj.
All right.
Spend some time
Thank you. Those are good questions. Yeah. Thank you, Melia.
Yeah. Okay. Thank Melia, Juxi. Thank you. So we have three other audience only want to ask a question. We can. So next one will be Dr. Du Guangwei. You can unmute yourself. Yeah.
Yeah, actually,
Hey,
very, very nice talk. Yeah.
Thank you.
So I have two questions. The first one, actually, I never really care about nutrient, but actually, recently I started thinking more about nutrient because I'm getting a little older too. But so from what I heard, usually DHA is good, right? So we are promoted to eat fish oil or all those kind of good things. But from your work, actually, incorporation of DHA seems really bad for aging. Can you reconcile what's going on here?
Yeah, there's a really confusing concept about what we take DHA for as a supplement, healthy supplement, versus the DHA content in cardiolipin. Remember, our body never trusts what we eat. Okay, if we eat meat, it will be digested into single amino acid. We do not use the meat protein to build our body. If we take a fat, it will be triglyceride will be chopped into free fatty acid and monoacid glycerol, or even free glycerol, then resynthesized. So is DHA. When we take DHA, it does not go where mitochondria is. So DHA is used for cell membrane and many other stuff. Actually, if you take DHA, at least in one study, it prevents cardiolipin oxidation. DHA get into PC, PE, PI, PS. Those are other phospholipid which serve as the bulletproof part to protect DHA to be being integrated into cardiolipin. Only in cultured cell, it will cause apoptosis because the DHA will get into mitochondria. And the DHA cardiolipin is very bad. It triggers oxidative stress and apoptosis. That was published quite a while ago. But it's a good question. So when we take a DHA, actually it's protective to our heart because it will improve the level of good cardiolipin, which is the tetralanolin cardiolipin. DHA cannot be easily get into mitochondria. And if it does so, it will not get into the cardiolipin, it got into other phospholipids.
So just to follow this question, because then where this DHA come from? Is there a different enzyme that's a remodel fatty acid in the mitochondria?
My understanding is that DHA is mainly from supplemented food. I don't think our body synthesizes DHA much.
That's what I thought.
Yeah. Just like omega-3.
我在听这个报告呢。
All right.
Yeah, and the second question I have is actually you have two enzymes. If I remember correctly, it's TAZ and ALCAT1.
Yeah, tafazin enzyme. Yeah.
See, they choose different substrates, right? They channel it to different actually.
Yeah, tafazin is an amazing enzyme. Actually, very few people pay attention to it because tafazin, my lab, because this seminar is already way too much information packed into 45 minutes, 50 minutes. So we have several papers on tafazin. A couple of they published, but the drug treatment. So naturally, you want to ask what happens to tafazin mutant mice. Those are Barth syndrome mice. What happens if you knock out the ALCAT or you give a ALCAT inhibitor? The amazing thing is that if you knock out the ALCAT or give them dapagliflozin, those mice will not develop dilated cardiomyopathy. So currently we are in discussion with the Barth Syndrome Foundation. My lab actually is fortunate being sponsored by Barth Syndrome Foundation for the last 15 years because those kids really are dying without effective treatment. And we presented our work very early on. And the Barth Syndrome Foundation really feel excited that probably help us to develop a dapagliflozin for Barth syndrome treatment.
So do you know how two different substrates are channeled by two different enzymes to make a different action?
Okay, so this two enzyme actually has no comparison. Tafazin is a mitochondria specific enzyme. It's inside the mitochondria, and ALCAT is the ER associated. It's actually at the ER mitochondria associated membrane. That's where ER and mitochondria contact. And for my next talk, I'm going to talk about the another enzyme we call LPGAT and the phospholipid transport from ER to mitochondria, because this is a very hot topic. Once again, Peter Walter, the lab clone, the Hermes complex. I think Ganzhen Ji is not here. He should follow that because the mitochondria behavior is controlled by ER mitochondria contact, ER lysosome contact, ER lipid droplet contact, ER Golgi contact, nuclear contact. So because mitochondria contact is very important for its function. So for ALCAT, actually, it was when we tried to publish, same as the PERC cloning, we did not want to bother to submit a big journal, mainly because we involving extensive revisions. And when I was in a company, I mean, it was Nahum Sonnenberg. He was the editor-in-chief, and each time I met him or emailed him, he would say, "Hey, Roger, why in the world you submit your PERC cloning to MCB?" I said, "You are the editor-in-chief, of course." He said, "No, that should not be the reason." I said, "The reason is that if we submitted a big journal, I probably lost my job because it would require years of work, to resubmit." And at a company, so that's the reason. Anyway, so this work, the ALCAT work, also we submitted to JBC. And JBC had a hard time to accept it. They say, well, cardiolipin is inside the mitochondria. Why it is remodeled in ER? I said, that's what we saw. And now we understand that the ER is the the fuel depot. ER is the gas station. That's all the lipid being stored in the ER. That's where all the acetyl-CoA is synthesized and stored. So ALCAT, after remodel, through the Hermes complex being transported to mitochondria. The Hermes complex involved in the Hermes complex has not been identified in mammalian cells, and we are very close in getting that. I think we get it. We just didn't have the resources to move forward. So long answer to your short question. The two has no similarity. One is in the ER, one is in mitochondria. And one is the acyltransferase, one is the transacylase. One is the physiological, one is the pathological. So the two really, and by knockout of ALCAT goes sky high because it promotes cardiolipin oxidation and development of cardiomyopathy. Okay?
Thank you.
All right. Thank you. Next one will be Dr. Guan Dongxie.
Hey. It's very nice talk. Just so much of information. The first probably ambition, you mentioned the ALCAT1 is enzyme. When you knock out, actually I probably missed the biochemical part. So this enzyme, the major function of this enzyme is relieve the ROS induced oxidation.
This enzyme itself does not cause oxidation. By changing the structure of the 4 fatty acyl chain, now cardiolipin is full of a double bond. This is a good question because most people get confused when I talk. I'm glad you asked it. So the more double bond the cardiolipin side chain has, the more vulnerable to get oxidized. If you store oil in your kitchen, if you store saturated oil, you never go bad. If you store oil which is very polyunsaturated, go bad very quickly. So it's like peanuts. If you buy peanuts, you store it in the room temperature for half a year, it tastes very bad. What happened? The double bond get oxidized. Now, because cardiolipin is localized in the site where ROS being generated, it's like me next to Chernobyl. That's where all the radiation. So I get radiated very quickly and die. So here you have a polyunsaturated fatty acid enriched phospholipid sitting next to the place where ROS being constantly generated. It got oxidized. The more double bond, the more free radical, easier it gets oxidized. That's how ALCAT promote oxidative stress by changing cardiolipin become polyunsaturated. That's the only reason this enzyme did so much bad thing. And this enzyme do so in response to the oxidative stress associated with aging and age-related metabolic disease. That's the whole mechanism of action.
Okay.
All right.
All right. And I have another question. So for your so you generate this inhibitor. Did you how you evaluate the activity of enzyme when you treat the inhibitor?
This is a drug development issue. So remember, in my first life, I have 2 lives. My first life, I spent 11 years at Eli Lilly. That's where I learned all the tricks, how to develop high-throughput screening, how to do a drug screening, how to do the structure activity relationship study, how to establish cell-based assay, and all those things that all come together handy when I come back to university. I cannot imagine if I didn't go to Eli Lilly, if I didn't spend 11 years there, I have 0 chance to get this far in drug development. So I cannot explain clearly within minutes how this assay is done. but it's basically take a lot of experience to complete the assay itself. Okay. In the past, we used the radio immunoassay to analyze the enzyme activity. Now we can use the fluorescence labeled substrate.
So usually, I'm not sure because just one small molecule you treat an animal. I wonder what's the phenotype going if you treat this same molecule to the ALCAT1 knockout mice in also the phase in like aging model or some other models. Not just check the that's not the direct target of the ALCAT1. I mean, whether have other unknown target.
Oh, okay. So you are asking whether if we treated ALCAT1 knockout mice with this compound, whether you will have any effect? Kind of whether it depends on.
Oh, you mean there is an off-target effect?
Yeah, that's probably your question. Yeah.
Yeah. It is difficult to address in vivo. Well, because if we use an ALCAT knockout mice, we give a compound. It's a negative because it's already protected from all the disease. Now you give a compound, so it is very, very difficult to address. If ALCAT is a bad thing, if it is a good thing, now you knocked it out, then you can test it, right? So
no, I'm not. Because for example, you gave the for generally speaking, you for example aging, right? You just delay the time you get that, not forbidding the phenotype to happen, right? You knock out the gene. So it's not the negative and the negative, right?
Yeah, if you give a knockout mice this compound, you can rest for sure there will be no phenotype because the enzyme is no longer there. The compound is not going to do anything. If you want to do toxicity study, you will do it in the wild type mice. You can give this compound with high dose and to see whether there's any liver damage, there's any other damages you can analyze. That's what we did with the CRO company, commercial company, and also by a big pharma and our own lab. So we did all extensive off-target studies. Also, if you look at this, let me switch to this figure. This figure will speak very loud about off-target activity. If it does so, it will most likely to inhibit two closely associated enzymes. LPCAT is about 40% homology with the ALCAT. Okay, so you have 100 micromole dapagliflozin. It didn't do anything. So if you have enzymes so closely related, and it didn't react with your compound, that's pretty supportive, right? The compound is pretty clean. Yeah.
All right.
Okay. Thank you.
All right. Due to the time, I think then we'll have last question from the Yue. I'm not sure whether Yue is still available. Her question is there any human data for DAPA treatment? Is it, or maybe non-human primate? I guess you guys you tried that in Hangzhou, right? For the using monkey to try this drug. I was thinking that when you were accompanying.
Yeah, we try to raise money to do the monkey study. So far, because biotech venture is really in trouble. Most biotech companies struggling. The VCs has not got their money back. So it's a very bad time to raise money to do expensive studies. If we have advanced this compound to human, as I said, I'll be the first one guinea pig. I'll be drinking teas with DAFA supplement. I have 100% faith on this compound because if I look at the mice, I feel that I wish like the mice. They are very old and they have such a healthy phenotype. So once again, it's a money issue. The company has survived the last 8 years, and finally it closed end of last year because next stage you require somewhere like $100 million, raise $100 million to move to human clinical trial. But if we have some rich friend in the audience, please help us. I'll be the first one to be the guinea pig. And
yeah, I think
going on with our primary. Hello?
Yeah. Yeah, I think right now in China probably is a good timing to do the monkey study. I heard that right now it's much cheaper because a lot of companies raise monkey for the price is much cheaper before several years ago. So
yeah, I visited Yunnan, Yunnan, what is that? Kunming, yeah. I visited Kunming Primary Center. Yes, now it's much more affordable. You're right.
Yeah. Actually, I have just one more question, short question. So you talk about the senescence, right? Right now it's a hot topic, cellular senescence. So what do you think about the whether it's really important because people talk about cellular senescence is important in the immunology and in young people, cellular senescence is even beneficial than what do you think whether it's really exciting or whether we should work on that or it's actually just not that important. I just do you have any suggestion for young investigator about the senescence?
Yeah, my personal take is pretty negative because there's a company called Unity Biotherapeutics. It was co-founded by Jeff Bezos, who is a billionaire, trillionaire, and WuXi Pharmatech, a bunch of Bay Area scientists and Mayo Clinic scientists, Judy Campisi and Jim Kirkland, Dao Hong. The company at one time was $2 billion. They were testing BCL2 inhibitor as a senolytics to treat age-related diseases. The company's stock used to be like 200 something. Now is $1 or $2. Every clinical trial has failed because the BCL2 inhibitor is the chemo drug. And BCL2 is not the cause of aging, okay? I personally don't think cellular senescence is a big deal.
All right. Okay. I think
it's the price we pay for.
Okay. Yeah. Thank you. Thank you. 现在我看了，现在那个我们这个seminar already last 1 hour and 45 minutes. 45 minutes. Yeah.
因为这个实在是不太好，因为什么呢？国内大家睡觉的时间，这边也不是 happy hour 的时间，因为这边是早上不能够喝酒，不是 happy hour。所以以后这个时间是不是还是要改一下？因为主要是
这次我们其实已经改革了，提前了半小时，因为不然的话，我们这边这次国内的，然后我看好多人来了，提前半小时多来了不少人。然后那个因为也考虑到，因为还考虑到东海岸那个 City of Hope 那边，文栋老师今天早上应该起了大早上是6点多。
文栋老师起得很早，是的。因为众口难调，
对我们是很好的。不过之前是跟陈真，我们是看看哪个时间段会更好。
今天我们提前了半小时，我觉得还国内参加的
会好一些。
然后那个
半夜太难了。
众口难调。
对，这个 City of Hope 是几点？现在是
我们现在
9点。
9点，对，咱们开始的时候是早上7点半。
那也差不多。
7点半可以的。
7点半可以，对。
再早一点，其实7点也可以，但是我觉得，就看这个 West Coast 有很多7点，他们可能就不一定会参加。
早上7点对吧？那肯定是不行的。
对，我当然 OK 了，但是我觉得7点对于很多的人有点早。
没事，我觉得还是可能7点更好。
7点吗？是国内这样是11点。
对，国内
那美东时间是10点。
10点。
很好很好。反正我们先试个11点半，现在还是现在这个时间提前半小时还蛮好的。
其实今天除了做seminar，我还是想跟大家一起探讨一下，就是中国的科研导向问题。我是觉得这个 CNS 其实给中国人挖了很大个坑，就是这个梦想基本上把你所有的 innovation 基本上全都没了，因为你要 CNS 必须是热点的，这个是我想给大家提醒一下的，就是说，就是我觉得可能我用这个图来说吧，就是说，比方说你老去追 CNS 的话，你一直给这个就是叫什么 Grey Master 打工的，他们把这个湖里的水全放干了，然后你给他帮他把这个鱼卖，每一个 pathway，他其实这个 Grey Master，比方说从所有的这些诺奖的，基本上都是把湖水放干的，就是他们的东西在他前10年没有 impact 零。就比方说像这个每一个领域在它最早的时候没人关注的，等已经有 impact factor 的时候，你就已经给它在填坑。这当然了，就是这是我一言堂了，我就希望有更多的人参与这个讨论。我今天这个老冯啊，这些都可能都在忙 grant 呢，没见。这个我是更想跟讨论一下，不光是科学的问题。
我们还有机会，
对，
还有机会再讨论这事情。
是，然后那个你们不是有那个 winter retreat 吗？不是应该可以在那个
老姐夫呢？没事，还有机会的，包括 annual meeting 啊，都是我觉得是可以多讨论这方面的。
对。然后那个我们可能搞一个这种special section 吧，这种 career development 啊，或者是 publication 啊，我们可以讨论一下这些 topic。到时候可以设置。
因为我想强调的一点，就是因为我去了意大利已经断奶了，给我原来的这个实验室彻底断奶，让我可以放飞，这样我才做了一些非常原创的东西。如果说我有 pedigree， 我只能证明我老板的工作更伟大，我一直接着做，明白吧？所以这个意大利10年让我变成一个就是一个野人。在一个荒漠求生，我到处挖坑，挖了坑以后别人帮我填。这当然了，就是 perk 这个东西，别人把它提前就把它给包装给卖掉了，这我也是挺可惜的。我是没想到那么快。Anyway， 这是我自己的感受，就是说你一旦追踪热点的时候，很难长期能固定到一个领域里面做自己感兴趣的东西。
下次那个代谢红论，您要讲什么？你要讲那个 LPCAT 吗？代谢红论。
这个华硕类就是 mitochondrial behavior 这一块儿，其实
红论你不是还要
研究 aging 的话呢，绕不过来。对，就是这个 H factor 的那个，就是 H 什么，那个叫什么，就是
H-top
代谢红论嘛，代谢红论就是红原那个代谢红论。对，我可能会谈一下这个另外一个课题，就是也是一个没人关注的，就是phosphatidylglycerol， 就是磷酸甘油酯的这个它的功能和它的重塑和它的运输，就是也是没人，就是因为科研在选美嘛，就是越人多的地方越人多，越没人的地方越没人。其实这刚好是和我的性格相反的，我不愿意扎堆儿，所以一直在做一些另类的。
那我插一句啊，卓贤今天辛苦了，感谢您Dr. Shi。我们之后再定好时间，文东我们可以再
你赶快休息吧，
我们可以讨论。
不是我要去 UCLA 参加一个 Mitochondrial Symposium。OK，
好
，OK。
所以您今天先给我打了个基础，能听懂更多。
Mitochondrial，
那是Orion 新人还在那儿
，Orion，对，他 organize， 昨天就开始，昨天我们就去了，但我今天就特意早上先 sign in 一下，怕有什么问题。
对，
昨天辛苦了，你那边特别晚了。
对，Orion 一直
不辛苦啊，
他比较有个性的，也是在研究。好吧，谢谢大家，这个今天实在是太晚了，
谢谢
休息啊。 ，All right，
没事没事，好，
谢谢
谢石老师，
谢谢卓显，谢谢孟老师，谢谢
不谢，
再见，
应该的，我们经常联系，
好的，那我们今天交流就到此结束，好吧？
好的，
谢谢文东，all the best，
谢谢 Roger，拜拜，
拜拜。